Okay, so we have a question. Charles, do you see that question?
Yeah, about Cinnabar. I'm gonna do it right
Uh,
now, real quick, and see if I
okay.
can get something.
So as far as using screenshots, this is for Danelis. You need to work that. I don't know if what Micah sent you, but it's not okay to use somebody else's work unless you've worked something out with Micah directly that it's okay to do that. So that's, you need to be sure about that first. Yeah, I mean, Valeria, sure. I mean, I don't mean to be dismissive. That's not my intent. We're all a little swamped. If you really, really need to see what went wrong with quiz 3, I'd be happy to schedule something. Um, at this point, it's not going to change the grade, and nothing that's on quiz 3 is really going to be repeated anywhere else. But if you want to, that's fine. Okay, Anya, so that's a little unusual, but let's assume that the information is not on Flybase. You still have other databases on which you can look up what SS is. I mean, I find that a little weird. I can try to help you with that or one of the other TAs, but you do have other options. Flybase isn't the only database that would record, like for example, the NCBI, PubMed. You might want to do a Google Scholar search or other searches to try to dig to see if you can find some information regarding your mutant. Okay, I find that odd because I know CPI is on there. Let me look. And if it is the case that the actual mutation hasn't been characterized, you need to write that, but you need to document that somehow. Oh, sorry, I need to admit people. So this is for Casey Hernandez. The figures that you need to include have been covered in the rubric and in the previous Q&A sessions. Do you have some, I mean, I don't quite understand. So yeah, you need to include your amplicon, your alignments. Do you have a, I don't know if Charles can see that question or Micah can see that question. Do you have something more specific?
Hold on, let me take a look. I'm actually looking at Cinnabar right now.
Yeah, I was actually trying to do Cinnabar as well, or in
Oh,
SF.
okay, okay. Yeah, no, I'm looking at it right now.
Sorry. Hi, Micah.
Hey, Mike, can you hear me?
Yes, yes, you're coming in loud and clear.
Awesome.
So I looked up, let me see, what is this? I'm trying to work on, I think Charles is doing Cinnabar. I'm doing SS1. Let me look.
Take care of him.
Okay, so whoever posted the question about SS1, there's literature on this mutation. So if you can cite the literature as far as you can, like for, I haven't looked at the literature, but if in the literature it says it hasn't necessarily been characterized to a particular level, then you need to address that in your paper. Does that kind of help with whomever asked that question? I'd have to scroll back up. Okay, good. You guys can type out your questions, but you're also free to, you know, speak up.
I have a question, if that's okay.
Sure, of course, that's what we're here for.
So I followed Ricky's bioinformatics video at first a while back, and then last night I followed Charles' bioinformatics video, and for some reason I can't find the amplicon for Brown whenever I put in the forward primer and the reverse primer, and I use the reverse complement. I couldn't find it within the get decorated fast A, and I don't really know why it's not showing up for my complement Brown mutant. Yeah.
Okay.
Yeah, I'll take a look at it. Just give me a sec.
Okay.
Hey, Danellis, to answer your question, all you need are your mutant sequence that we give you in your FASTA files, right? So the mutant sequences that are on Blackboard, you can just blast that against a wild type version of your gene. You don't technically need the amplicons. The last email I got from you was like 2 weeks ago, I think.
So, but he's answering your question now, though, regarding that was Danellis, right? Who just asked the question about Ricky's. Okay.
Yeah, the last email I got from you was, I said, could you send them to me? Each of the sequences and primer combinations is a bit of a story. And I think that was 4-16, so that was right before our first Q&A, or I guess our previous Q&A.
So how about this? Let's just work it out here. I mean, we have Micah present, Charles present, me, Casey, Sarah. So do you want to ask your question one more time, Danellis, and then have Micah repeat the answer? Since he's here, he can just address your question now.
Oh, okay. So I sent another email on the April 20th. I don't know if you received that one.
Okay, but can you please ask your question? So Micah might look for the email.
Okay.
But yeah.
Okay, so I tried doing the amplicon for both Cinnabar and Scarlet, and both only give me the forward primer and the reverse primer.
So even if you don't get that amplicon, you can still blast your mutant sequence that you got from Blackboard onto a wild type sequence of the gene that you can get from Flybase in BLAST, right? So the only reason we have you find that amplicon is so that you can get the wild type sequence of the gene for whatever portion of the gene we want you to get it for, right?
Okay.
So if you can't find your amplicon, then you can use Flybase to just get the whole sequence of the entire gene and then blast your mutant sequence against that wild type sequence if you're having trouble finding the amplicon. And then I sent you the answer to Scarlet. Like, let's see. I sent both you and Vinesh the answer to your Scarlet mutation, basically like the result that you should have gotten. And I'm looking at the email right here where I sent it to you. It looks like a blast result of 2 separate blasts because Scarlet would give you 2 separate alignment results.
Okay, can you show me really quick how to get that whole wild type sequence that I can compare with my mutant sequence?
Yep.
Thank you.
Can I go ahead and share my screen real quick?
Absolutely, Micah. Do what you need to do.
All right. Share. Flybase. All right. So if we were going to Cinnabar, right, we would type in CN, press go, and it would take us to the Cinnabar gene. And then all you do is press get the sequence of, you can do the gene region, which is a whole bunch. You don't really need that much, but you can do it if you'd like. Or you can do the extended gene region or the coding sequence. Any of these 3 are really going to get you what you need. So let's do coding sequence real quick. You press get sequence, And here's the sequence of the wild type gene. So with your amplicon, the point of the amplicon was for you guys to be able to get a portion within this gene that you're going to be looking at. Now, if you can't get your amplicon, then you can just use the whole gene to move past that, right? Because if you don't have that second primer for some reason, or it's just really not working for you and you're running out of time, then you can just use it. It's okay.
Yeah, I'm actually having trouble finding the second primer also. So.
Yeah, the amplicon is more like an extra step of work. If you absolutely can't find it, that's okay. Just explain that you weren't able to do it, and these are the reasons why. Your second primer wasn't working, and that's fine. Boom, answer right there.
Okay, so then I compared that whole wild type with my mutant sequence for Cinnabar and Scarlet, and then I should get the mutation on the figure like Charles showed on the video.
It's not going to be exactly like Charles showed it on the video, because Cinnabar is a deletion mutation and Scarlet is a transposon, right? So I believe Charles' video, he found like an actual mutation, like a base pair change or deletion or insertion or something like that. But for Scarlet and Cinnabar, you're going to get two blast results. One is going to have it above where the mutation was, one's going to be below, but you're not going to be able to get in between because your mutant sequence doesn't have that in between area.
And
okay, I'm sorry.
I was going to say, that's okay, Danellis. I was going to ask Micah to explain again the idea of the transposon and how that's influencing the result. If you could expand on that a little bit.
Okay. So transposon is a jumping gene, right? And we went into this like in one of our lectures. Basically, you got a piece of DNA that comes from some other portion of the genome, and it comes in to your gene based on these inverted repeats right here. These inverted repeats will match up into your gene, right? And it inserts itself in, okay? And that ends up ruining your protein, your end result, okay? So maybe it messes up with the promoter region, maybe it messes up with the actual gene itself. In any case, your gene is no longer expressed properly.
So in their searches though, this insertion prevents them to match things up in certain wherever this is inserted. That's
It prevents you guys from matching it up, right? Now for a deletion, whenever you have a huge span of DNA that's just completely gone, what you're going to do, or in some cases what you'll find, is that you'll have a piece of your mutant sequence from the beginning matching up and a piece of the mutant sequence from the end matching up. But the wild type sequence that has that deleted portion in it will not match anywhere to your mutant sequence. So you'll still get two results, right? It just is going to take you explaining that in your results and discussion, what's actually happening and why you're seeing this with your blast results, right? So all of your results technically have already been discussed in another paper. You just have to go into Flybase, figure out what the results were from previous papers, and from the data that you gathered in this class, you just verified it. And that's it.
That's right? Very well. That's good. That's a good explanation, because I think the one thing I want to emphasize from what Micah said is that you guys are not making any novel discoveries, which is an advantage, right? You could look at the literature and it'll describe what your mutation is. What you're doing is verifying that, as Micah said. So okay. you already have the answer. So go ahead, Charles.
I figured out why Cinnabar is not hitting. It's because there's a freaking mismatch in the primer.
Oh yeah, that was Cinnabar, the Cinnabar primer.
Yes, second.
And
Cinnabar
I believe
I
sent that to your group, Danellis.
Yeah, my thing is I cannot find like the mutation in the chromatogram like Charles did, so I cannot like pair that peak with the letters, you know. So then our figures.
Did you check the dot plot? Because so if you have a deletion, which is what Cinnabar 1 is, so I did two things. The first thing that I showed you was how to look at deletions. And for deletions, if you're lucky enough, you'll catch it in your sequencing data. If not, then you won't see it. But if you do see it, when you do the dot plot, you should be able to see a big chunk that's missed, like that's not there. So I kind of did go over how to do deletions. So actually, if you go to, because if you're looking for Cinnabar 1, it's a deletion. So that's what you should be looking for. But then again, it's also 1.8 KB. So maybe
Yeah, so you're not going to find a tiny little area like Charles did.
exactly.
It's not going to be like Charles, right?
Oh, okay.
It'll be different. And so you just explain those differences based on the previous results.
Yeah. So what if you do a dot plot, and what you'll see is possibly just the first part match up, and then suddenly it just stops matching up, and everything else will look different. So you'll see half of the dot plot pretty much.
Well, the thing is, when I tried to do it, it was just a straight line. I couldn't see any like you showed. It didn't show like that. It was just a straight line.
So the reason why it's a straight line there is because it's not one or two base pairs, and BLAST will treat it as a completely different alignment, right? So it'll give you the dot plot for that alignment. But because there is such a huge mismatch, such a huge gap, it's going to only show you the mismatches within that one little bitty alignment. So if we go over here, the results that I sent you were like this. This is for Scarlet, I believe. And so see how we have two here?
Yeah.
The results, right? So you've got one at the beginning and one at the end, but because of that deletion or whatever this one is, actually Scarlet's insertion of a 412 transposon, then you only have two matches at the beginning and the end.
So it's okay if I use that same screenshot because since I cannot get there.
Yep, that's
Thank
okay.
you. Okay.
I gave it to both you and Vinesh and anybody else who's emailed me.
Okay, thank you.
I think
Yep, no
it's because I did mention that you had to approve that, Micah, before you joined. As long as if you're using that screenshot, that's fine. I would just simply add that you still have to explain what you're looking at.
Yes,
Right?
yes, you do have to explain it, right? So that's just a figure, right? You need to explain everything, give me a nice figure legend and everything, because I literally just gave you a picture right there. So you need to tell me what's happening.
Okay.
Okay, those are good questions from Danelis. So Micah and Charles and myself and Sarah and Casey, maybe we could put together or just say now what would be some common types of mutations that students may encounter. Like we in lecture we talked about insertions, deletions, transposons, those kind of things. So is there something that we could maybe go over quickly just to say, look, if you line it up, you'll see this or you'll see that. That would, because everybody's going to see something different depending on their mutant. Do you think that might help?
Okay, let me
Not necessarily the basics, like, because I think they understand what an insertion or a deletion are. It causes, but like within the context of some common mutations that, like for example, I think it was Cinnabar that was a transposon. Was it Cinnabar?
No, Cinnabar 1 is it's a deletion.
A deletion. Okay, and then which one's the one that was a transposon we were just talking about? Is
Scarlet.
Scarlet. So there we go. So what I'm saying is, for example, Scarlet would be a transposon, which would cause a certain type of mutation. And then when you look at it, like you were saying, Micah, you're going to see parts of the gene match up, but not the middle because it's all messed up because of the transposon, right? That's what I'm saying. If we could kind of just reiterate those type of results. If they have
actually, I'm trying to get some like real results right now that I have on hand just to show what an insertion deletion change, something different is going to look like.
So that when they're on their own, they can say, okay, if this is what I'm getting, then this is what it might be,
Let me see. Give me a second.
Yeah, take your time. So is it Anya Gull says, our bioinformatics figures are 2 amplicons, 2 DNA sequence comparisons, 1 protein homolog, and analyzing bad reads like in Charles's video. That seems a lot more than what I've had in mind, but certainly you're going to need your DNA alignments, your protein alignment, and then I don't know what you mean by bad reads, but I think that the middle part, the DNA and protein, I would think that that's.
Yeah, so the bad reads would kind of be the DNA alignment, because when you do the alignment, you're looking for the mismatches or whatever mutations you're looking for.
Yeah.
And And the interesting thing is, even with the what you call the bad reads or the looking for mutations, if you do find any mismatches, you know, that could be novel and interesting too. Because most people report on like the functional stuff, right? But there could be actual SNPs in there that, you know, could be interesting. So it's okay to bring those up too.
So while Mike, I don't know if Mike is ready or not. The one thing I want to make sure students understand is what are the absolutely required figures for their bioinformatics, and what are options?
Okay, optional is a picture of your protein. Required, you have to have something between your protein and a protein of another species, right? Humans, bugs, whatever. That is absolutely required. You will 100% for every single group find that. You must have an alignment, a DNA alignment of your mutant to your wild type gene,
okay.
Right? You should have an alignment of your complement to your wild type complement. For the amplicons, you should technically have 2 amplicons. If you aren't able to find it, I think that Danellis was saying she was having some trouble with Scarlet. You need to tell me that you are having trouble. Don't put it. Okay, if you have a reason why you aren't able to find something, don't just not put it. Okay, the only time I'm going to take off points for it, if you tell me that you didn't put something into your paper because you couldn't find it, is if somebody else was able to find it. All right, if somebody else was able to find it and you weren't, there's an issue.
All right, if you guys want me to go over Cinnabar, I can go over that real quick. So there's a couple of extra steps that I took along the way to get to find the parts for Cinnabar.
All right, feel free to do that, Charles. You can share your desktop or however you want to do it.
Yep. Yep. Okay, cool. Got it. All right. So let's see. Share screen. See if y'all can see this. Can you guys see this?
It says you are, yes, we can see it now.
Okay, cool. So I will start from scratch. So delete all this. So this is Cinnabar. This is the forward. This is the reverse. So I'm gonna look for those sequences. So if you did it my way, it actually was not gonna work. But let me go over why. So what I taught you guys to do was to get the declarative FASTA. But actually the reverse sequence is not on here. It's actually going to be further down. So what you're going to do is go 5,000 like base pairs, like north and south of the sequence. So you just get more sequences around your actual gene. And only by doing it this way, you'll be able to find your reverse primer. So copy that, paste it, and then highlight everything. So this is all just according to what I did. The only difference so far is just adding the extra base pairs north and south, upstream and downstream of the gene. And then you're going to remove all of the spaces. So place all. Yep. And then let me make that bigger. So probably better. All right, so one of the primers you can actually find on here. In fact, if you did it the other way, you would be able to find the forward primer. So let's see. Where is Cinnabar? Oh, there it is. There we go. So there's the forward primer, and you can tell it's a protein coding sequence. It's the pink area. And then if you do the reverse primer, and this is actually kind of why it didn't work. So I'll show you the process through. Copy this. I'm actually going to blast this. So home, blast. This is the result. If it takes you long, I'll skip over to the results. Let's see.
So all we're seeing on your screen is your find and replace. I don't know if you've moved on to another screen.
Oh no. Okay, so you guys, did you guys see me find the primer?
Yeah, I think that's where it stopped, sort of. It seems.
Okay, so I might have to, yeah, so it's actually not sharing my blast with you guys. So let's see. How do I share my blast?
If it's on your screen, it should. Is it underneath this window, or do you have two monitors?
No, it's on one monitor. You guys can't see
Okay.
it right now, right? This is I'm
No.
showing the blast. Okay, so I think I might only be showing you guys screen. Okay, maybe this is better.
Yeah, I think you may have selected a window instead of a screen.
Okay, that's probably what I did. Can you guys
Yeah.
see my blast?
Yeah, we can see
Okay,
it now.
so pretty much what I did was I took the reverse primer, which is right here. You guys can see me like,
Yes.
okay, circling. Okay, cool. I just went to Flybase, I blasted it, and if you look at the blast results, you can tell one of the nucleotides is actually mismatched. So if you search this, this primer, you're not going to find it because it's going to mismatch right here, and it's going to give you an error. So you can find this, and you should be able to find the end part. This part you're not going to be able to find. So, so what I would do is, again, you still have to do the reverse complement. So actually, I'll do it again. But copy this, reverse complement it, submit. So here it is. But as you guys know, like the first part and the end part don't really match. So you can just copy the first couple of nucleotides. Go back to here. You guys can see my Word document, right?
Yes.
Cool. So this should work. So there it is. And then we'll just find the end part. So right here. There it is. So there's your second primer. And it's actually going to be, again, a little further, I think, up or downstream of what they normally give you from the decorated FASTA. So that is your, let's just underline that, I guess. And that's your amplicon for Cinnabar. So it's doable. There's just, I guess, 2 extra steps. One is because there's a mismatch. Your reverse primer, you're going to have to look for the beginning and the end. And the other step, I guess you guys can't, you guys can see me do this part. So I'll quickly go over this again. But let me go back here. So normally I go to get decorated FASTA, but this doesn't show enough base pairs around Cinnabar. So you just do like 5,000 or maybe 2,000 will even work. But this will just give you more bases north and south of this sequence. So you just have more bases to play with around Cinnabar. Yep.
Nice job.
Hope that made sense. But yeah, so yeah, one primer is on the protein coding sequence. This part is on an intron. Yep.
Yes, thank you very much.
Oh, actually, let me stop sharing.
Okay. Thank you, Charles. All right. So we can take more questions. I see we have a little bit over 20 people in the room. So feel free to ask your questions. This is you have all of us available.
And the other one was brown, right? Brown
Yes.
didn't work? Okay, let me take a look at brown real quick.
Who was asking about brown? Maybe if they
I asked about brown. So I ended up, so I couldn't find any matches with the Finch TV. I couldn't find it with the primer forward and primer reverse, but I was able to find it with the wild type BW. So wild type brown brown forward and reverse. So I don't, there's like 2 files and I don't really know what.
So you're actually looking at the Finch TV files, right? Not the actual amplifier.
Yeah, I'm looking at the 6 Finch TV files that I have.
Okay, cool. Let me take a look.
It's group 19.
Group 19.
I don't know what other info.
It's okay. I'll look for it.
And also from digging on Flybase, I found that it mentioned a 412 inserted transposable element.
Yeah,
in
those
the
are
location. So like, does that mean like at that specific 412 location that something was inserted, or?
So it's the 412 transposon. So it's that's your jumping gene that actually jumped into your gene. So generally, working with transposons is pretty confusing. Oh, are you in Micah's class?
No, I'm in Casey's.
Casey's what day?
Wednesday too.
Wednesday 2, got it. And group 19, okay. And then
So I couldn't find the amplicon with the forward or reverse. So then I did the thing that you mentioned earlier with getting the sequence through CDS.
right.
And then that one worked better.
Okay.
I just had to do like a lot of combinations of things.
Yeah, exactly. It's once you understand how to utilize all the tools, then it's kind of just mismatching things here and there.
Okay.
Like for example, I showed you guys how to blast it against a known sequence, and that kind of gives you the dot plot. But you can actually blast the sequence by itself in Drosophila, and you won't get a dot plot, but then it'll find your thing. So if it's actually transposon sequence, a dot plot wouldn't give you anything.
Okay.
But okay, so I'm actually looking at Group 19 Finch TV, the folder. Which files did you have troubles with again?
The first two, brown
Brown with
primer forward and brown primer reverse. Those two I wasn't able to find anything.
I think those are with prune or
Prune?
P. Well, I don't know what PR is.
Wait,
actually.
yeah, let me look.
All right, here, let me share my screen. Maybe Michael will know.
Okay. Because I just figured PR meant primer, but I guess not.
No.
Oh, no.
It should be
Purple.
So,
That's purple.
yeah,
probably.
it's
Yeah, PR is
purple.
purple.
Yeah.
So you're actually looking in the purple gene. That's probably why you didn't find anything.
Okay.
But it
That
should be
makes a lot
wild
of
type.
sense.
Brown with a purple should be actually this one, right?
Yes.
So brown with that should be
Yeah,
purple forward and
that's
purple
purple.
reverse.
Because in my group we never worked with purple. We only worked with sepia and brown, to my knowledge.
See.
So then I would just ignore the top two then?
I mean, yeah. I mean, there's really no point in looking at purple and brown. So the story that you would tell would be, okay, this is is sepia your complement?
Sepia is my mutant.
Oh, okay, great. Then
Yeah.
then you would be able to find your mutation in here, right? So
Mhm.
whatever you find in here would be your actual mutation. And then wild type brown, actually that wouldn't show you anything either, because that would be a wild type fly looking at the brown gene. Which should
So
be,
I did do that combination, and I did it with both files against the Flybase CDS
Mhm.
reference from earlier. I was able to find some things, but like one of them there was one nucleotide difference, and then the second one there was 3.
yeah, so
I'm like
that's fine.
confused.
So those could be novel SMPs, but they might be like silent mutations. All right, they
Okay.
could be silent, or they might even be inside of a non-protein coding area, so it doesn't really affect anything. But it still could be interesting and novel to look at those things, for whatever reason, right? So you can still bring those up. I will. For me, I would be okay with that. And I don't know, is Casey in this?
She's here.
She's here, but she said she wasn't gonna be able to talk. Remember?
Oh yeah.
Yeah,
That's right.
that was yesterday, guys.
Oh.
Oh,
Yeah,
okay.
I'm fine with that, and I just want you to be able to explain, like they've said before, explain what that means.
Okay.
Yeah, because obviously the wild types don't have any mutations in them, or they don't have a brown phenotype, right? Which means that if there are any differences in the brown gene, it's not causing a phenotype. So yeah, definitely just say that, even though there are mismatches against the wild type sequence, those could be SNPs that are silent.
Okay.
Yep.
All right, I think that answers my question. So like which version would I use then, since I did them both? Would I use both of them in the paper, or?
I mean, the most important one would be the sepia keel, but I mean, I don't know, but if Casey's okay with it, I guess you can talk about those things since you've done something, and those are kind of cool figures. It kind of shows that you've done the blast.
Okay.
So figure-wise, I would focus on the mutant. Just make sure that you include that.
Mhm.
But if you want to do it as supplemental, then that's fine. But that's just not what's required.
For the complement, you mean?
For the
Okay.
complement.
Okay.
So for your actual mutant, you want to make sure that you include that.
Yeah.
Mhm.
Okay. All right. Thank you.
Yeah.
Okay, we can ask somebody else have another question. Anybody else? Scarlet. So Charles, I think it was, I'm sorry, was it Michelle that was just asking the question?
Yes.
Okay, so because I went and looked into our sort of our master sheet. It has, so your mutant is sepia, right? Sepia
Sepia.
teal.
Yeah.
And then your complement is brown,
Yes.
right?
Mhm.
Okay. So Charles or Micah or Casey or Sarah, would that, I mean, if, did you get to do your complementation crosses?
Yes.
You did, okay.
Mhm.
So would, I mean, because that's part of the reason I'm bringing this up is because that might explain why she has those primers in her group, right, of the
Right.
FISH files.
Right, but normally you would look at the sepia gene in brown to make sure it's wild type. So you would look at your complement gene in your complement, or whatever. So you, for example, you'd look at the brown gene in sepia, and that should be wild type, and that's kind of your control. And then you would look at the sepia gene in brown mutants, and that should be wild type. So those are the controls that would normally be there. But brown with purple really would not tell you anything because you're not even interested in purple. And also with wild type flies, I guess I take that back. With wild type flies
Yeah.
and on the brown gene, you can confirm that the wild type, well, because then what Michelle found would actually not, because those are technically missense silent mutations. So it
But
wouldn't
it
really tell you, you know.
should tell her something though. I mean, if she is able to look at the brown and wild type, right? About something about her mutant.
Yeah, you would confirm that your wild type flies are
Right.
indeed wild type for brown.
Right. Yes.
I guess
So
so.
it's
That
not
would
brown.
be a control, you know.
Because I, you know, wondering if she could use those for anything in her paper.
Yeah.
So,
But she did find mismatches.
Yeah.
So so one of the things you can look at is you can try to figure out if those mismatches are in a protein coding or non-protein coding region. If they're in non-protein coding, then that's an easy way to say they're novel SNPs that people probably haven't found, but they're, you know, but they're not contributing to the protein at all. That'd be in a, you know, perfect world, I guess.
okay.
But
All right.
if you find them in a protein coding region, then it could be, you know, on that wobbly base on the third, you know, the third base of the codon. So, yep.
Okay. Thank you.
Thank you.
So while we're waiting for more questions, so I'm not sure. It's almost 3:45. This is gonna, Micah. What did you have in mind? Is this just going to be a free question
so
and
I
answer,
had,
or?
yeah,
Go
kind of free question and answer, and I'll stay here for a while. But we had a question a while ago to see different results of a blast analysis for different scenarios. Do you want me to cover that real quick?
Yeah, sure, absolutely.
Okay. So let's see. Share screen. Share. Alrighty. Let's see. So I just pulled a random sequence off my computer a while ago. I did a base change, right? A base change mutation. So I switched like a C to a G or some other base to some other base, right? So just like you guys, I ran my mutant sequence that I sequenced in at Lone Star Labs against my wild type sequence. Right, so my mutant sequence is going to be on top. My wild type sequence is going to be on bottom. I always put my wild type sequence on bottom just so I don't get confused. Let's see. So this is actually the mutation right here. So I switched. If this is my, let's see, my mutant, and this is my wild type, then my mutant is going to have a G. My wild type is going to have an A, right? So I switched it around. Some of you will have those base changes, right, in the paper, you need to write the difference between a transition versus a transversion. If you have one of these where it changes from a C to a G or a C to a T, you need to tell me if it's a transition or a transversion. If you guys have something like a 1 base pair deletion, then you'll have something that looks like this, where you get these little dotted lines or these dashes, right? So let's go down here. So this section right here, see how that's a dash right there? So that means there's a 1 base pair difference there, which means you have a deletion, a 1 base pair deletion. A 1 base pair insertion would be flipped, so you'd see the dash on the other side. And then for those of you who have really, really big insertions or really, really large deletions, you'll get something like this. So this is a really large deletion. All I did was take a wild type sequence of an integrin, and then I deleted out like 1,000 base pairs or something like that. So what I got was 2 alignments. Okay, so see how this alignment goes all the way up to 960. This one starts at 961, but this one starts at 2,161. So you see that there's actually a big gap in between because of that large deletion that I have.
Do you have a dot plot for that? Yeah, so you can even show that on the dot plot. There's like a. Yep.
And then let's see. All right, yeah, that's pretty much it.
Okay.
So for the insertion, you'd see the opposite of that. So you'd see a large increase, but you would still get these 2 results right here.
Excuse me, can I ask a question about the large scale deletions?
Yeah, what's up?
How would you show that in the paper? Because I know the video said you would like align it to the Finch TV sequence.
Yeah,
Like,
so
insert-
for my students, if you want to include the dot plot, that would pretty much tell you whether or not you have a deletion or not, right? Like that's pretty clear. So if you want to do that, that would tell you have a deletion. Or you can include both.
Yeah.
Yeah.
That would be a really good visual way to do it. You can also write about it as well. Doing both is probably the safe bet.
Yeah.
However, if you feel like you just want to write about it, or for some reason you can't get the dot plot, then just write about it.
Right, thank you.
All right, good question. So I think there's a new question in the text, right, from Casey. Everybody see that?
What does it say?
It says, when we are looking at this alignment tab, would we check the mutations in Finch TV to make sure it is actually a mutation?
Yeah, always confirm against a chromatogram to see if it's an actual a confident read or just a bad read.
And so, Charles, could you just very quickly go over what they might look for to see if it's if it's actually a mutation or something just a bad?
Yep. I can pull that up on Finch. There's a good demonstration of that in my video,
Yes,
but
there
I'm
is.
gonna go over. Yeah, but I'm gonna go over that real quick, just to show you guys what a bad read looks like and a good read. So this is I guess sepia with sepia primers. So let me pull up my video. Share screen. So there you go. So that's can you guys see the chromatogram?
Yes.
Okay, great. So as you guys know, you always want to remove the beginning because that's bad, right? And then you always want to remove the end because that's bad. So so man, this is actually bad overall. So overall, this is pretty bad, and I'll kind of explain why. As you can tell, there's a lot of like these small peaks that are underneath. Can you see this? I'm kind of highlighting this, right? There's like a small blue peak right here, which is like a C, which is underneath the T, right? So that if this is a mismatch right here, even though the T says it's a confident T, which is probably going to be right, but let's just say you get a mismatch at this T position, right? And you can have a C, it could actually be because this is the actual read underneath it. if that makes any sense. Or for example, if there's a mismatch here, this A and this T, and they're flipped or something, right? Like I would like one, notice how low these bars are and they're underneath this dotted line, which means the confidence at these two positions is not really high already. So if you get a mismatch at this position, then throw it out because it's not really usable. Another example is, for example, let's just say these are good reads, right? They're not great, but just say this peak is, it's a nice clean peak, nice clean peak, nice clean peak, nice clean peak. Then at this position, notice how the bar on the T is lower. And it's probably because you have an A right underneath it, right? So this is not really a confident peak either. So if you have a mismatch at this position, you know, it's kind of sketchy. I don't, yeah, it's maybe not the best idea to keep this peak and say that it's a mismatch. This might actually be just because it's a bad read at this position, right? You have some overlap, a die blob or whatever. So when you do include the mismatches, I always tell my students to include the chromatogram right above it and make sure, you know, so if you have a mismatch in the blast, make sure you have nice clean peaks or nice large tall bars right above it, showing just that it's a confident read, but it's still a mismatch.
All right. Did that help?
Question?
Okay. Well, in any case, that was a good explanation.
Mm-hmm.
Okay. Anyone else? How's I mean, we have a few students in the room, of course. So how is everybody doing in terms of the evaluation forms? Any issues there? Any problems? Is it working pretty well? Anybody want to chime in?
I had a question about that actually. In the lecture video, you mentioned about how if everyone in the group gives everyone 100 and how, you know, that works.
Yeah.
In my group in particular, we all worked really, really well with each other and we had no issues and we were all on time with all of our crosses. So how would we, I guess, you know, be able to give each other like our evaluations without seeming like we are just giving ourselves 100?
Sure. So if you look at the evaluation form, it isn't just a across the board 100, but there are certain sections that you have to look at. I'd have to bring up the form because I don't want to misinform you. Let me see if I can't bring it up. But you know, I'm not going to say that your group members didn't deserve 100. The point in my video is that it's very unlikely that everybody gets a perfect 100. So that's why I'm asking that you kind of be fair, but nobody's perfect, right? So I think the areas are time and effort, and then there's 3 subsections for involvement. And you know, if you really think that all your group members deserved 100, then okay, but that's very unlikely. There's always room for improvement. And I mean, there's always room for improvement as me as a coordinator and instructor, right? I could always do things better. So does that kind of help? And I mainly said that because in the past, when we get these groups that all give each other hundreds, it's usually because they're not really being careful with the form. They're just filling it out to get it out of the way.
Gotcha.
So I hope that helps. Again, I'm not saying that you and your group members didn't deserve hundreds, but you can understand why I have to be cautious about that. Okay. But I do, you know, kind of maybe think about how things could have been better. I'm sure I'm sure you could tell me things I could do better, right?
Because like part to me is like, okay, it was like, for example, like something small happened. I was like, I don't want to nitpick and take off points for small things. So I'm just sitting here like,
yeah,
do
that's
I
up
take
to
off
you.
points for that or not? Yeah, okay.
That's up to you. I'm not going to, I mean, it's not like I'm going to deliberately look for people that did this. It's just that, for example, last semester, this happened where I started looking at evaluations and group after group after group were just giving each other hundreds. And I'm going, that's just can't be the case. And this is a little difficult, right? Because we're doing evaluations kind of thinking about each other. So it's kind of a hard decision I have to make about what would be normal, right? And it goes the other way too, if you can think about it. If there's people that are just completely giving everybody else bad scores. That's
Yeah.
also unusual. So I have to make some tough calls, but generally it'll work out. Don't worry too much about it. Just be honest about what you're doing, and it'll be fine.
All right. Thank you.
Do any of the TAs have anything to add about that? Any wisdom?
Just give everybody 95. Just kidding.
There you go. Okay. Oh, by the way, since we mentioned evaluation, so the instructor evaluations are also available. Did you guys get emails regarding that? The students? Anybody?
Oh yeah, I got an email.
Right, but the students for genetics,
Oh,
they're the
Yeah,
ones
we
filling
did.
out. Okay, all right, great, because those are done online. It'd be great if you guys could take care of those and you know, give us your feedback.
oh yeah, I read all the comments too. So if you guys want to just have at me in the comments section there, I'm ready for it.
But it's anonymous, right? So
Yeah,
so
it's anonymous.
Micah
So
doesn't
you
know who
can,
wrote the comments.
yeah, you can just take a baseball bat and break my windows.
Okay. Okay, so Sarah took care of an answer there, a question. So far, we've had some really good questions that cover some broad topics, and this is being recorded. I can see the recording light, so I will post this. I will, you know, render the file, give it to Micah, and Micah will put it up on YouTube. Okay, so anybody have any more questions? By the way, when we have these downtimes, if any of the TAs want to chime in with any concerns or advice, by all means.
I would not wait until the last day to get this done.
Yes.
Yeah.
Very good
I
advice.
would definitely, I've had a couple people ask me like, feasibly, how long is this going to take? And I'm just going to tell you right now, it's not a question that you should ask.
That's
it will take as long as it takes to get it done. I've seen people work weeks on this thing, and I've seen people do it in 2 days and fail. I would not hold it off, for sure.
good advice.
And every day that you don't start it is just another day that you lose. So, Dr. De La Flor, what about the test? How's the test going to be? Tell us about the test, Dr. De La Flor.
Okay, so I'm currently working on a test, but not the genetics test, because I have two screens. But no, in all seriousness, what I can tell students that are logged in right now is that, first of all, it's probably going to be 50 questions, right? Pardon me, it's probably going to focus on the bioinformatics. There will be some basic genetics and fly genetics. It'll be online, it's open book. You'll have 75 minutes, I believe is what I gave you guys last time. So that's about 1 minute and 10, 13 seconds or so per question. You'll have 24 hours in the sense that it'll be open for 24 hours, and anytime during that period, you'll be able to take the exam. And I believe I've already scheduled it, but I have to see, I'd have to look at the syllabus. When did I schedule it for? To look at the syllabus.
I think it was May 6th.
Yeah, that sounds right. So I did get a couple of emails from students asking, you know, why it had been pushed back. Well, because of the delays in spring break, right? We got pushed into another, we lost a whole week, and then trying to get everything online, we lost some time. So it is within, usually we don't do it within the finals week. Unfortunately, this time it's kind of bled over into that week. I don't have any other choice. So that's all I can tell you about the exam right now. As I write it, and if I feel that I need to do this, I will put up, I will send out an email with more specific information. But you know, I don't think it's going to be that difficult. If you understand the bioinformatics, and by the way, understanding bioinformatics doesn't just mean knowing how to do your blast searches, which is important, but you need to understand the concepts behind that, the concepts behind DNA and proteins and their structures that allow you to do this kind of searches. So that's important as well. So some very basic but important molecular biology. which is all covered in the slides, by the way, that Micah and Charles put out.
Eric asked, will there be a reading assignment 5?
No, it's only reading assignments. I didn't see that. So usually we have 4, well, not usually, because this is a new thing I've introduced, but there are 4 reading assignments, and there's not a 5. So the photos, Micah, how is that coming along? The bonus point.
Oh
photos.
my goodness. Oh, it's going, do you want to, okay. Am I allowed to show some of the submissions?
Sure. I mean,
Because-
does any, any of the other TAs, can you think of any issues? I can't think of any issues.
Like, is it absolutely, like, some people went above and beyond.
Show
All
some.
right. Absolutely above and beyond. Okay. Well, while we're waiting for questions, let me pull it up on Blackboard real quick and make sure that everything's safe before I actually start sharing my screen.
So while you're doing that, I just want to make sure that, first of all, it's open and people are submitting so it works, right?
Yeah.
So people,
Yeah,
so, 'cause that's-
open submissions. Submissions are great.
While you're
Let's
doing that,
see.
again, I'll recapitulate that our bonus points for this semester are mainly going to be the reading assignments that I've put out and the photo stuff that Micah takes care of. Those are, because we can't do any of the crosses, right? Because we went online.
All right. So,
Ah,
we got some nice ones in space. Looks like this one is the beach. All right, there we go.
Hey.
Oh, we got one in a test tube. And Drosophila is the student. Okay.
oh, that's really cool.
Oh, we got another one in space. I've seen like quite a few in space, and some of, oh gosh,
That's
this one's
nice.
probably one of my favorites.
Okay,
Goodness.
I recognize some of the faces. It's like,
Yeah, this is in my class.
nice.
Let's see. Oh, this one's
Ah,
okay.
that's me.
Okay. Oh, hey, there you are. Yeah. Okay, so we've got another one from the same group. Okay, we love genetics lab. Stick figures. I respect the stick figures.
I
can
like it.
respect the
I like
stick
it.
figures.
I wish I was that thin.
Oh, we've got a heart.
There we go.
Yes.
This is great because it's I mean, it lets us know students have a good attitude.
Yeah.
Even
Wash
with all
your
the
hands.
yeah, wash your hands. There you go.
Okay.
Nice. Oh my
Ooh,
god.
Okay. That's an interesting one. All right. We got a frog eating them up. Oh,
That
another
was
space one.
Baylor College of Medicine. Space.
Oh, some more space.
So while you're doing that, each student, like so for example, in a group of 4 students, they do one photo and then they submit that same photo, and each one gets their 2.5 points for this.
Yep.
Yeah,
I'm not making
right.
them submit multiple photos. It's
Okay.
fine. All right. Hey, this one, I'll accept this one. It's in a beach. Okay. I see the beach. That's fine.
That looks like a Zoom background.
Okay. So then we have something like this. As long as they edit it, it's
Yeah.
like ever so decent. I'll go ahead and give them the points on that one.
Yeah, no, yeah, of course. They
Yeah.
submitted.
We got some of this. Oh, this one is very, the quality's not so great, but oh well.
That's
Okay.
that's a fly pad, right? They're all on a fly pad.
Yeah, they're on a fly pad.
Yeah.
We got some more beach. Oh, we have one person in the chat.
Okay.
Did you say PowerPoint was a way to do these animations? You can use PowerPoint. There are online softwares as well, like literally for free. You just go on Photo Editor. They have some really, really great, easy things to use. We got some more space.
So just while Eric, this is to Eric. These aren't animations. They're just photos. They're still.
Yeah, just photos.
Just to clarify. Hey, that's cool. Wait. Go back to that one. Go back to the fruit fly one. So
Okay.
I should use this as a question in the exam because all of these are male fruit flies. And I see.
Oh,
two
yeah, they
female-
are.
I see two- Anyway, good photo though.
Let's see. Oh, we got, oh, see, this is a nice one. This is a nice park area.
Yeah, it's nice.
I went ahead and gave them this one because even though it's in the Fly Lab, they did put themselves in space, or rather they put space in the Fly Lab. So what did I comment on?
You don't know. I
The
mean, that
galaxy
could
in the
that
Fly
could
Lab.
actually be happening in the Fly Lab. You know, just
That's
happened
true.
whatever. That's
Quite possibly. Except I think I may have been the one to take this photo. So, oh, this is interesting.
it's a dungeon?
Are they actually at a place? No, I think they're just all.
Yeah, they just dropped themselves in there.
Oh, well, that's cool. That's actually pretty well done.
Yeah. Nice.
Wow. I'm gonna put that one a 2 right now. Okay, this is decent.
Yeah.
All right. At first, I thought this was an alpha helix, but then I realized it's confetti.
You thought it was like like a protein structure, huh?
Yeah. Okay, on the beach.
Nice.
I like it. Oh, we got the heart again. All right. We
Fly
got
pad.
space. Oh, the Power Rangers.
Mm-hmm.
See this one? This one disappointed me. 0 points for creativity, 0 points for paying attention, 0 points for making me smile, 0 points for being original. But they still got 2 points for this.
That's right. Good.
Okay, washing your hands again, always important. All right, in space. Okay. Wash hands.
Yeah,
got
so
that
we're oh, that's a new one.
Oh yeah, and we're slowly running out of groups.
Yeah.
All right. Well.
Yeah, that's good examples, and that should give students. I mean, I don't know how many have submitted. But
Yeah.
I encourage you guys to tell your group members, or if you know the groups, to submit their photos, because that extra 2 points or 5 points can make a difference.
Oh yeah.
Oh, they like that one.
This one.
Spider-Man.
Oh my gosh! Week 2 of quarantine. Sunday, Monday, Tuesday. Oh, it's all the same. I get it.
Nice job, though. Nice job.
Yeah, this is nice. Okay. It's interesting how this Spider-Man is facing forward, but the head is facing backwards.
It's Spider-Man.
Oh, we got some more space. Frog. This one. And last one is not here. Okay. Download submitted file, maybe? Ah, okay. I'll accept it. It's there, technically.
Okay.
I've never seen anybody submit something like this, but that's cool.
As long as it's there.
Yeah. All right.
So we're down to about 12 students, and I'm subtracting the TAs and myself. About 12 students, I think, 12, 11 students. So you guys, do you have any other questions that we can address for you?
I'm doing something on the side. I will ask the question in a little bit. Sorry about that.
Okay.
I mean, I got time. I've got Netflix here.
Yeah, I mean, the only limitation, as I mentioned, Micah, is the file size, because I am recording.
Oh, yeah.
I'm recording. So I think if it's okay with you, I'll till 5. That's 2 hours of recording. That'll be a huge file.
Yeah, that's fine. That's fine. I mean, I'm okay no matter what. I got some cookies. Got a piece of bread.
Okay, I have my question.
Okay, Michelle.
So before I mentioned that I did both Ricky's and Charles' bioinformatics. So I just did Charles' version of the bioinformatics for my sepia mutant sepia, and I'm getting 100% matches whenever I'm trying to find the mutation. But then on Ricky's, I was able to find an insertion or a deletion. And would it be okay if we use, for at least for our figures, one of them be Ricky's format and then the other one be Charles's format?
Yeah, whichever finds you results.
Okay, because I was wondering if that would conflict with it being like uniform or like the same format type of situation.
Well, maybe keep your figures looking as similar as possible, or try to keep the same formatting in terms of how you set up your figures. So if you're using one color for whatever, stay consistent or whatever. But other than that, I don't, yeah, just whichever gets you results.
Okay. Like just, I guess the formatting of the page results showing like that itself is different, but in terms of like showing what's where, just keep that.
I think that they went through quite a few different examples for you. And so there's so many different approaches to how you can get to your answer. But whichever ones you feel are going to explain your information, your data the best, then you can do that. And then as what, like he was saying, like just being being consistent with the way that you put it in your paper, in your figures. But, there's so many different multiple approaches you can make toward getting the same thing. So whichever one works for you.
All right, thank you.
So Michelle, since you've got a captive audience, and if you're comfortable, you can always share your screen and your TAs can look at it right now and tell them.
Yeah.
Oh, I'm using my laptop for Zoom, but then my computer for, because my computer doesn't have a microphone and whatnot.
Oh,
so
okay.
That's why I can't share.
All right.
All good.
You can always send it to me in email too, Michelle, if you want me to just take a look.
I was actually thinking about that.
Okay.
I will do that. All right, thank you.
So let's see. See if I can think of anything to. I have the WA5 rubric up, and I'm see if anything sticks out as far as something to talk about. Because I mean, the last Q&A session that we did, Micah was pretty detailed, went over everything. Does anybody have any concerns about the rubric? Any questions? This has been pretty much bioinformatics focused. If not, that's okay too.
Yeah. Anybody have any other questions? Like live questions? Really, any question to get us back into talking about WA5 with the class? Maybe like a break question?
Break question? Like what?
Like just if you're feeling stressed, you got to take a little break. Like
okay,
sometimes when I have like a long hour lecture, I'll break like 30 minutes in and start talking about something completely different.
Ah, I see.
Yep.
Okay. Are any of you taking summer courses? Any of the students?
Oh, that's a good question.
I don't know yet because I don't know if it's a good idea or not, considering if everything is still going to be closed. Because if everything's still closed and we're all still stuck at home, then yeah, why not, right?
Mm-hmm.
But then,
so
yeah.
Classes will be online for the summer.
Yeah.
Because I'm teaching one course in the summer, and I've already got students signing up, but it's all online.
Genetics lab?
I'm sorry? No, it's
Well,
not. No, it's not. There's no genetics in the summer. This course is only offered in the spring and fall. But I'm teaching another course, another biology course.
Okay. Because I have something planned over the summer, and I don't know if it's going to get canceled because, like, right now they're like, "Oh, we're gonna reopen," but like, are we gonna close again or not? So it's all up in the air, I guess.
Yeah, I normally teach A&P over the summer. As far as what we're doing, I know that the class is going to be held, but for what the TA involvement is going to be like, I'm still kind of unsure.
So I have a couple of, I know a couple of graduate students that are teaching that right now. And so everything's online. So the lessons, the exams, quizzes, because you guys have weekly quizzes, right?
Yeah.
It's, as far as I can tell from what I've heard, everything's online somehow. Dr. Gill has managed to put everything online.
Oh, wow. Yeah, he's actually really good with that kind of stuff. He'll just crank it out like an entire class works of material.
Okay.
So here's a question for the students. Do you guys prefer the online format or in-class format?
Ooh.
Of which course?
In what way?
This course.
This course. Okay.
I don't like the bioinformatics online, personally.
Okay, and that's perfectly understandable because it's not easy.
Yeah.
Normally, I sit down with the students and we'll look at the data together on their laptops or whatever in class. It's easier to do that.
Yeah, it's 100 times easier, and it takes a little bit less time, and it's fun too. It makes it a little bit less scary. That's the kind of unfortunate part about you guys having to go through this alone.
I will say that you guys are doing a really, really amazing job considering how difficult this is with like all the Q&As and just all of the videos and whatnot. So I just wanted to give you guys thumbs up for that.
Well, thank you.
Thank you.
We
Yeah,
appreciate that. Okay, so Eric asked if I will upload or when I will upload, and it'll be later today if everything works. Because we did this. This is the third one, and the first time we did our Q&A, I think I screwed up somehow, and I didn't set the settings right, and it didn't record properly. So that one was just a live thing. Then we did a second one, and that one got recorded, and this is the third one. So it says it's recording. I can see the recording light on.
Yeah.
So I have I had a Zoom meeting with someone at the University of Mississippi this morning, and they were also wondering how they're going to be managing these type of courses over the summer, because there are, for example, biochemistry or organic chemistry courses, which are lab courses, like AMP also. And so he was asking me if we had figured out any way to do something like those courses. And the answer is no. I mean, because if for some reason, let's say fall 2020 comes along and we're still having problems with the virus and things are shut down, I'm not sure how we would handle this course, because a lot of this course, like the flight genetics, has to be all done in person, right? So we'd have to figure something out.
Yeah, this one is going to be a little bit of a tricky situation if this actually starts extending.
Right.
Especially for, I mean, I may go with computational work. Like my PI is like literally this close to saying, okay, Mike, it's time to start putting computational work into your proposal.
Yeah.
Yeah, that's what I've been doing the whole time, just computational work.
Okay.
Have you guys ever tried kombucha?
I don't know what that is.
I've heard of it, but no.
It's like, okay, so my fiance, she got this stuff called kombucha, right? And I honestly don't know what it is, but it's pretty decent. It's like a really sour carbonated drink.
Where do you get it?
If you don't have, like Target or Kroger or Walmart, as long as you don't get the ones, or I don't get the ones with like sugar in them, and so it's like super strong, and it just kind of like punches you in the throat a little bit. Yeah, it's decent. It's a decent drink. I can only drink a few sips at a time, but when I do, it's a good time.
Are they? I mean, do they have a lot of calories or anything like that? No.
It's 30 calories.
Okay, that's not bad. You know what I don't like about these calorie labels? Sometimes you pick it up and it says, oh, it's 30 calories, but then if you don't look closely, it's like, oh, that's 1/4 of the bottle. It's 30 calories, If you drink the whole thing, it's not 30 calories.
Okay, so let's see. It says amount per serving is 30 and 2 servings per container. So I guess 50 calories.
Yeah, so Eric wants to know if it's an alcoholic drink.
Yeah.
Oh, is it an alcoholic drink? Is that why we actually have to get carded for this? Because it's alcoholic drink?
This is why Micah is so happy right now.
Okay, let's see. Pregnant or breastfeeding women should consult their healthcare professional.
Oh my goodness.
Wait, no. It says verified non-alcoholic. Okay.
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 I'm gonna have to put a disclaimer on this video. Oh my gosh. Okay, guys. Well, we'll sit here until you, I mean, this is a good time to try your bioinformatics. We're here. I mean, Micah has said he'll stick around at least till 5. It's around 4:20. So
4:20.
you guys have us captive, so it's a good time. So, but we are losing students rapidly. We're down to maybe 8 students.
13.
Well, yeah, but you got to
How about
count me and 4 TAs.
Oh yeah. All right. How about this? If we don't get a question within the next 10 minutes, we close up shop.
Okay.
Yeah, that sounds good.
Okay.
Yeah.
All right. Well, I'm trying to think of things that I've covered that are important, but I've covered everything. I don't know if you guys, the TAs, have been watching my videos, but I try to cover everything like grading or schedules or things of that sort, the administrative stuff in videos. And I think everyone's pretty much understood what we're doing and the changes are.
Mm-hmm. Hopefully everybody has been reading the emails and watching the videos, or else they're going to be really confused.
Yeah, that's yeah. Okay. So then if we don't get any more questions, I'll call it a day at 4:30.
Yeah, that sounds good.
I will adjourn. So my
Do you have any plans?
Charles, are you teaching anything in the summer?
I applied to teach something during the summer because I'll be a TA this summer, but I just don't know what I'll be teaching. I mean, last summer I got RA, and the summer before that I was teaching ANP. So I have no idea.
Okay. Yep. So you asked do we have any plans? You mean after this?
Oh yeah.
I think if it's not raining, it's cool outside, I'm gonna go do a run.
Oh yeah, I did actually, like right before this, I was going to be like incredibly late, right? Like absolutely, I might have just missed the whole thing if Alexa hadn't like called me and said, "Hey, you want to go to Michael's?" And I was like, "Well, can't go to Michael's because I got somewhere else to be that I just remembered."
So I think Eric is asking a question. He says, when you say grading, did you discuss how grades will be broken down, or is it normal grading? No, that hasn't changed, Eric. The range in which something is an A or a B is still the same. The only thing that changed, and I went this in detail, is that I had to remove one quiz. So there's a little bit, instead of 1,000 points, I think it's 986 points that you guys can score, plus whatever your bonus points are. And that really doesn't change anything, because instead of dividing your total points by 1,000, I'll just divide it by whatever the available points are. So it's still the same. It doesn't change anything. I did get a little bit of pushback on that. I think a few students didn't understand. But so nothing has really changed the way things are graded. I hope that helps. So that brings up a good issue, though, and I had planned to talk to someone about this, but it is my understanding that some students will have the option to ask for a pass-fail. TAs, anybody, instead of being graded?
I think so. Something about that, but I think they have to do something before that. The Rintu Couture.
Yes. So I'm going to ask about that.
How do you spell her name? Renu Couture.
Yeah. I think students will decide by May 8th which one they want. One is they get, I think it gets factored into their GPA, the normal grade, and if they do a pass-fail, then it doesn't affect their GPA.
Yep. The only thing that I would say there is, for those of you who are looking to go into medical school, I don't know what they're gonna look at. I don't know how they're gonna take that. Medical school or any other professional school.
If you do just a pass-fail.
I honestly have no idea.
So Noor Khan has asked a question. You want to take a look at that?
Oh yeah, I have a figure of the two flies next to each other, mutant and wild type, that I got points taken off for. So will I get points taken off for it again on WA5? I can't take a new photo, so what should I do? Noor, you're not in my class.
So whose class? Who's your TA, Noor?
Why did you get points taken off for it?
Yeah, that's a good Charles. Hey, we have Charles.
Yep, I'm right here. Let me take a look.
See, this is what a Q&A is supposed to be, right? We have the TAs present, we have students, you can ask questions, you get immediate answers.
So most of the times I took off points, mainly because the pictures could have just been cropped better. So if it's a quality issue, then it can't really be changed. But most of the time it can just be like cropped or adjusted later, like post-production stuff. So let me take a look.
What if I didn't get any pictures at all? The only pictures I have used in WA1, but they weren't the best. I didn't use any at all in WA3. From Casey Hernandez. Casey, I remember grading your paper. Honestly, you need to, I don't believe I took anything off there. What I took points off for most likely were your figure legends. Because I knew what state your group was in. You guys did a really good job of communicating with me. Each person of your group was telling me what was happening. So I don't believe I took any points off for that.
Casey, does that address your question?
No, I don't. No, I understand that. I mean, what if I don't have any pictures for WA5? Oh, so if you don't have any pictures of your slides for WA5, you're okay. You're still fine. Put what you have from WA1, but you're still going to have other figures, right? So you'll have your chromosomal diagrams. You'll have some data tables with whatever you have. You'll have your chi-squared tables of however far you got. And then you have your alignment and your amplicons, all your bioinformatics stuff, and then maybe a protein picture if you want. If you guys still have time, I just wanted to double check that I'm looking in the right place for human homologs, human diseases, and my mutant gene. Like, if I am looking in the right section on Flybase, is it okay if I can use my mic? Yes, you can use your mic.
You can also share
please
your
explain.
screen if you want.
Yeah.
Okay, hello. So I'm just checking from Flybase. I'm the same person. I don't know if you guys remember from last time. I said my gene was white and I couldn't find any human homologs, so I was doing something wrong there.
Oh, I remember that.
Yeah.
Didn't Charles show you how to do that in the last one?
Yeah. So I was digging around here. Also here on Flybase, when I click on human disease associations.
Yeah.
And then it takes me to this website and it gives me references. Is this the correct website? I don't know if you guys can see on HCNC symbol report, and then it gives me a list for the APC transporter. I could just screen share if,
yeah. You can screen share. It's okay.
Okay.
Who's your TA?
Casey.
Casey. Okay, I believe she's still here. So.
Yeah, I mean, I'm just starting. Oh, sorry. I don't know if you can see.
Yes, we can see your screen.
Okay.
Do you want to step back a few screens to show us how you got there?
Sure. Let's see. So I went on Flybase and I searched my gene, and just to see if I could get a bit more specific, if I could go to, oops, sorry. If I could go to, sorry, maybe a mess, human disease associations.
Right.
And then so I click on the one with I click on this first one. So would it be correct for me to go from this website and then here they start listing some references? So would this be like some good place to start? I was just.
Yes, Casey, what you can also do is scroll up. So the approved name, copy that all the way over, all the way up to the first parentheses. So don't include the junior blood group.
Okay. So,
all right, copy that.
Okay.
And then put it into Google.
Oh, Google.
Okay, I see what are you doing.
and then press Uniprot, or type Uniprot on the end. Uni, and then PROT. Yep. And let's see,
First result?
ATP. Yeah, go ahead, try that one. I don't think it's the human one though. Oh yeah, it's human.
Yeah,
okay.
it is.
So now you can run down this entire page and it'll tell you everything about your protein. It'll give you the structure of your protein. It'll give you the important domains of your protein.
Nice illustration.
Okay. Okay. Yeah, I just wanted to check because it just gave me all of this, and I know you guys didn't mention. Sorry. You guys probably didn't mention it, so I just
Yeah, there's a lot of good stuff there. The only thing is it's difficult to understand because of the different resources that they come from.
So I know because my gene was white, I'm pretty sure like it probably the one that had like a lot of that was most understood. So I just didn't know why because for WA3 I started to try to find some human homologs, but I couldn't really find anything. But I think I was just Googling Drosophila white gene. Okay. Okay, thank you guys.
So you can also use BLAST to find other homologs in other organisms as well. Right, so you just take that entire white sequence and throw it into BLAST.
So okay. Sorry, this is my mute.
All good, all good.
But just take your time.
Okay.
Yeah, no rush.
No, because hang on. My computer's a mess. So yeah, I might have misplaced it.
So you can
do it right
find
now.
it if you want to go ahead and find it, because what you're doing here will be recorded and other students will benefit from what you're doing.
Okay. Okay. Yeah, sure. Okay. Sorry. So I would go to get. Sorry. Oof.
Yep. And then go ahead and copy that entire FASTA sequence.
So this is the white, well, the white wild type gene sequence.
Yes. Actually, make sure that you copy it without the numbers in there.
Okay.
There we go.
Back up. There you go. Oops.
Wait, sorry, I'm gonna.
Yeah. Take your time.
Okay.
All good. All good.
Okay, so.
And now go to BLAST.
So, if it, so we can.
Well, actually,
it was easier.
Go back. Type in NCBI BLAST.
Yeah.
So Flybase is really good at finding BLAST results for flies, right? Drosophila and Melanogaster. BLAST, NCBI BLAST will find it for everything. So go up, scroll up. You see nucleotide BLAST right there? Yeah, click on that. And then paste your sequence into that box.
The whole thing?
Yep, whole thing. And then you can press BLAST.
Also, I was trying to, because I got so I was following Ricky's YouTube tutorials, and that's how, okay, sorry, I'll bring that up later. Where do I go from here?
All right, so that's all of your matches. So you got the D-mail match, you've got, keep on going down.
Yeah, keep.
You got a couple of synthetic constructs, keep going down, down, down.
This is it. Unless you want me to go to, yeah, I think this is it.
Here, give me a second. Let me.
Yeah.
Let's see.
You can. Oh no, I was gonna say.
What's up with that?
There should be a few. There should be other. I mean, because this is an ABC transporter. You should have more.
All right. Let me try it real quick.
So I think Micah is gonna try to help you out here.
Okay. No, I wanted to bring it up because when you guys were discussing it earlier, I followed Ricky's tutorials around when they were posted, and I was able to post my forward primer and my reverse primers, and I was able to get the sequence. But I tried to do it right now today, and it gave me an error message. And
what gave you an error message?
NCBI, the primer blast.
Oh.
So I don't know if you guys want me to leave this screen because I really don't know what was going on. Because I was able to do it like fine a couple weeks ago, and I think when you guys were using it, I think you guys were able to use it because I have everything saved, but it just worried me that I couldn't do it again, which I thought was strange.
So Micah, do you want to kind of walk through that?
Yeah, I
So
could.
I'm just running through.
Yeah, I could
real
do
quick.
it real quick because I was able to do it really quickly.
So,
And I double checked these, and they were the ones. And I click get primers, and it gives me this error, and I don't know why. Because I double checked, and they were the ones that were on the Blackboard. So I don't know why I get this error message. I was able, like, I saved this like weeks ago. But I just don't know why it wasn't working for me again.
What are we looking for? Let me see if I can.
Yeah.
she's trying to look for homologs for white, and she was going through the steps of doing
Yeah,
it.
from Ricky's YouTube bioinformatics.
Let's see. Did you do your protein blast for that?
No, I haven't done the protein blast yet. I'm just following off.
I see.
Yeah, I think this is
So
it.
Oh, wait. Go back to that screen.
Which one? This one?
The one that you were just on. Scroll down a little bit.
Bioinformatics? Oh, this one?
Melanogaster is spelled incorrectly.
Oh.
Yeah, that's a typo that's been there before.
yeah, but the tax
Oh,
ID,
really?
tax ID is correct, I think.
Right.
Okay. Yeah.
It's
Yeah.
still the same.
So yeah, to answer your question, if you can't find the DNA homolog, or you can't find a homolog through the DNA blast, it's okay. If you do the protein blast, you should be able to pick up the human homolog.
Right.
Okay. The thing is, I'm like, again, because like my gene is white, I'm assuming I shouldn't have any like errors. So that's why I thought.
If you want, we can do the protein blast for white right now. So, yep. If you go to Flybase.
Okay. Flybase.
Yeah, you just the website.
Just the website. Just go Flybase.
And go to your gene. So white. And then on the right side, give it a second. It should pop up. It should be. Let's see. It should be like protein or let's see. G model and products. So yep, right there. Let's see, and then go to transcript data, or polypeptide data. So if you scroll down, it's one of the pluses. One of the, yep. And then okay, so on the left you see name and then white PA. Yep, click that. Perfect. And then let's see.
Oh, wait, you got to go to the RefSeq.
Oh, sorry. Yep. Not this. I go back to the Flybase page. Yep. RefSeq ID on the right.
Yeah. Right. So.
Sorry. Yep, right there.
Okay.
And then it should be run blast, right? Run blast on the right side. Yep. And then blast.
Okay. I was just wondering because I know maybe like not everyone in my I was just wondering like if everyone in my group they're going to try to do the very first step in Ricky's bioinformatics tutorials and they're going to end up with this error.
Yeah, check out my video.
Okay.
This part is at the end of my video, which is the protein blast. And is this all in Drosophila?
Yeah, I'm getting, because I followed that, and it's similar to the original results we got. It's all Drosophila.
When you hit run blast, you need to change the organism to Homo sapiens.
Yeah, I think that's.
Oh, we didn't. Okay, yeah.
Yeah. So.
Do I go back?
Yeah, go back.
Go back.
Just do.
There. Yeah, there. Type Homo sapiens.
Yep.
Thanks, Sarah.
This first one. It
Yeah.
seems like you spelled it correctly.
Yeah, the tax ID is all the same. Yeah.
Okay. So blast.
Yeah, this will give you your human one. Yep.
Oh, okay.
That's
That's
right.
a new species of humans.
Yep.
Okay. Yeah, that was just the issue because I already did it. Searching for the primers, like for me, wasn't a big issue, but I'm just like worrying why I had a D error message.
And there you go. Pick
Okay.
your favorite one, I guess, but ATP binding cassette.
Okay, and so it would be the best for me to just to select the one with the smallest D value, correct?
Oh, well, for my students, I told them whichever one fits their introduction, because if you're writing your introduction about a disease associated with one of these like ATP binding cassettes, well, I guess these are all the same gene, I guess, G, but yeah, whichever one fits your story.
Okay, because the problem was for writing assignment 3, I didn't even include a homolog again because I couldn't find anything. And for my writing assignment 4, I just selected a paper that I found, but I wasn't really happy with it. So again, that's why I'm just asking like, where can I find like the best results for me to like speak about a homolog? Because for my abstract, I just included a line about the paper. So I really just want to choose like a new one probably.
That's
So,
okay. I mean, is that
yeah.
okay with you, Charles?
Well,
my TA is Casey.
Oh, I'm sorry. I thought it was Charles. Sorry. I
Yeah.
apologize.
But yeah. So that's why I was just asking. But thank you. I think this mostly answered my question. But again, like the
yeah,
error
so
message.
yeah, so
I'll
for
follow Charles bioinformatics tutorial on that. I'll figure
it's at the end of my video to get to this point, but
it.
I
Okay.
remember one of my students before had white, and he was talking about like macrophages and blood cholesterol levels.
Mm-hmm.
So that was his story, and it had to do with one of the ATP binding cassette subfamilies or whatever. I don't remember if it was G or whatever. It might have been ABCG2, which is a little further down if you look at it. But you know, whichever one fits your introduction or story, I told him to use that one.
Okay, but just to be fair, so like Dr. Mike, it would be fine because I don't even remember what I mentioned in my abstract, but I could just pick a new one and a new paper and a new human disease, and it would be fine, right, for running assignment 5?
Since you didn't have a homolog in the
Yeah, I didn't.
and I wasn't a very big stickler about that, so
No, yeah, because I remember in the email you mentioned for me to find one. I couldn't find one that I was happy with, but like I knew it was weird like afterward because like, oh, we have white, so my abstract just found a random paper that, well, not random, but like a paper that was related with it. So if I wondering if it's okay if I could just change it like to something that I can.
Yes.
Okay. Awesome. Okay.
Great.
So is it okay if I stop sharing? Like,
Yeah. That's fine.
Okay.
I think we're pretty much done because we're at an hour and 45 minutes.
Thank you. Yeah, thank you so much for answering my question.
And I think other than you and some of the TAs, everybody's left. So.
Oh. Okay. But again, I'm just asking. So like that error message that you guys saw me had with my primers, is it because there's something wrong? Because I was able to do it a couple of weeks ago. I don't know if you guys remember.
I saw the error message. It looked like something with the website, but I can't answer you. I don't know if
try doing it Charles's way.
Okay.
Because Charles's way is kind of the manual way to do it, I guess. And Ricky's way uses a program.
Yeah, Ricky's way is sort of a shortcut. It's still all online. It uses the NCBI tools. He just uses them in a different way.
Okay.
Yep.
Yeah. So go with Charles's stuff.
Okay. Thank you.
Okay, guys. It's I think I'm gonna go ahead and wrap it up. It's 4:43, and we're really down to maybe 4 students. And
Sounds good to me.
Thank everybody for coming in, and all of the TAs were present, and I think we had a good session. And I'm going to go ahead and end the meeting, and it should render, and then I'll put it up. I'll send it to you, Mike. I'll put it up somewhere. You can grab it, and then you can put it on YouTube. Put a nice cover on it.
All righty.
All right, guys. Thank you very much. I'm going to hit end meeting.
Bye, everyone.
Bye-bye. Thank you.